Welcome to Unit 3, and in this unit you'll learn everything you need to know for cellular energetics.
Cellular respiration is a redox process, meaning that it's an oxidation process where we are we're breaking down the chemical bonds in glucose, because glucose is now our electron donor. And most of it is oxidation, where glucose would lose electrons. And these electrons will be transported to the electron transport chain. And finally, the electrons would be accepted by oxygen and reduced to form water. A neat way to remember it is just oil rig, oxidation is loss, reduction is gain. So here I am losing the electrons, and here water is gaining the electrons. So as you can see here, if I start breaking down glucose, okay, I will release electrons. So let's start out with one big molecule. Let's say that is a big glucose molecule. And all that energy is stored in the chemical bonds. Now it will be broken down stepwise. It's not all those chemical bonds are not going to be broken down all at once. It's going to be broken down bit by bit. So now I have two molecules that formed that still have chemical bonds, and it's still at a high energy level. But what I've released now is some of those electrons. And some of this energy I've used to make ATP. So yeah, and then it will continue. I will break this down even more in the Krebs cycle to ultimately end up with very low energy products. But I do all of that so that I can get those electrons. And the final electron acceptor is water that is reduced. Water is reduced because oxygen accepts those electrons. For cellular respiration, the core overview is we take these carbohydrates that we got from photosynthesis, and we need to slowly start breaking them down to release energy. Okay? Because cellular respiration transfers the energy from biological macromolecules to make ATP. Now, the first step is glycolysis. Okay? And glycolysis will happen in the cytosol or the cytoplasm of a cell. Then we'll have pyruvate oxidation, and this happens in the matrix of the mitochondria, where it will then enter the Krebs cycle, and this is where we would release most of the carbon dioxide. And then all these electrons that are being released will be transferred to the electron transport chain, and then we'll make ATP via chemiosmosis because of the proton gradient. So ultimately, we take organic molecules and we break those molecules down, okay, in the mitochondria to create ATP. Respiration happens in two places. It happens in the cytosol, and it also happens inside the mitochondria. Okay. Now, we've already learned that the mitochondria is also a double membrane structure, meaning it has an inner and an outer membrane. This is the inner membrane and the outer membrane. And then in between, it has something called the intermembrane space, which is very important. Okay. Now, glycolysis happens in the cytosol, and this is a shared common pathway for all organisms. Then the pyruvate enters the matrix and this is where it will be oxidized, and we'll learn what that means. So pyruvate oxidation happens in the matrix, the Krebs cycle happens in the matrix, and then finally, the electron transport chain and ATP synthase, it happens in this inner membrane. Okay, at this inner membrane of the mitochondria. So this is where we'll find the electron transport chain and ATP synthase. Now, if we look at the electron flow in mitochondria, okay, so this is the mitochondrial matrix. This is on the inside of the mitochondria. This is the inner membrane, and this is the inner membrane space, because here would be another phospholipid layer, and that will be the outer membrane. So here, the intermembrane space is very, very important, okay? Because this is where we'll get a high concentration of these protons. And it works similar to the electron transport chain in chloroplasts. So every time this electron moves, okay, it pulls in a hydrogen ion against the concentration gradient. So here I have a very high concentration of protons, and here I have a very low concentration. Now the NADH and FADH2, it got its electrons, it's carrying the electrons all the way from the Krebs cycle, okay, where glucose was slowly being broken down. Now every time a chemical bond is broken, it releases those electrons, which is then carried by these two electron carriers. So yeah, they carry it to the electron transport chain. The electrons move through the chain, okay? And then the final acceptor of the electrons here are oxygen. So oxygen will combine with two hydrogen ions to form water. So yeah, in plants, the first thing to donate electrons is water. And in respiration, the last thing to accept those electrons and form water again is oxygen. So oxygen is the final electron acceptor in aerobic respiration, and it combines with the electrons and the hydrogen protons to form water. Now, chemiosmosis, chemio just means chemical osmosis. So it is where we will be using a proton gradient to create energy. Now, what happens in both plants, in both chloroplasts, and also within mitochondria, is we'll have the electron transport chain. Now, as these electrons are moving through this chain, it actively pumps in more hydrogen protons from the matrix to the intermembrane space in mitochondria. And this creates a proton gradient. So here we have a very high concentration of hydrogen ions, and over here we have a very low concentration. And then mitochondria uses that to create ATP. So as this proton is flowing back through ATP synthase, okay, so as it flows back through ATP synthase, it drives the synthesis of ATP. So we phosphorylate ATP. Yeah, and that's chemiosmosis, and it happens in both mitochondria and chloroplast. Slightly different locations, but we learn. If you like that lesson, don't forget to like and subscribe, and I'll see you for the next one.